Namaskaram, my name is S. Ashok Kumar. Welcome to my channel. In the last part, I started explaining about the transport of oxygen. The same concept I am continuing with the oxygen dissociation curve. Actually, how this oxygen dissociation curve will obtain, it is actually a graph between PO2, means the partial pressure of oxygen, with percentage of hemoglobin saturation with oxygen. Actually, what is the importance of this ODC? Different types of factors like temperature, oxygen, carbon dioxide, how these factors influences the association between the hemoglobin with the oxygen. That means whether these factors, if they increase or decrease, whether they are influencing the association or whether they are influencing the dissociation between the oxygen and the hemoglobin, that we can clearly understand with this ODC. Before going to tell about how those factors influencing the ODC, first I will explain how ODC, standard oxygen dissociation curve will obtain. So I already said it is a graph between partial pressure of oxygen with percentage of hemoglobin saturation. Okay, for example, if the partial pressure of oxygen is at 10, then how much hemoglobin gets saturated? That is a 13.5. That gets saturated, hemoglobin gets saturated. Like that, if it's become a 20, the saturation will increase, that is a 35. If it is a 30 partial pressure of oxygen, it become a 57. The way of understanding you need to understand. If it is a 40, it become a 75. If it is a partial pressure of oxygen is a 50, it become a 83.5. If it is a partial pressure of oxygen is a 60, it become a 89. If it is a 70 partial pressure of oxygen, it become a 93. Then if it is a 80, then it become a 94.5, almost reaching to the saturation point. Then if it is a 90 partial pressure of oxygen, it become 96.5. If it is a partial pressure of oxygen is 100, complete 100, then the percentage of hemoglobin saturation is 97 or 97.5. That is why I said 100% of hemoglobin saturation is not possible. This partial pressure of oxygen, if you maximize this to the 100%, then maximum hemoglobin saturation is a 97.5. That means this partial, by using this value, partial pressure of oxygen and percentage of hemoglobin saturation, these are the standard values. Okay, that means at the standard temperature, that is a 37 degrees centigrade in our body, then blood pH 7.4. Like when you take the standard conditions or standard internal condition, based on that, this is a partial pressure of oxygen, this is a percentage of hemoglobin saturation. If you draw a graph between these two factors, we will obtain the standard oxygen dissociation curve that I am going to show that one. Based on the values I given before, if you draw a graph, you will get the graph like this. This is a standard oxygen dissociation curve. Here one important point for the NEET exam is, what is the shape of ODC? That is the one word you need to remember. The ODC is a S or sigmoid shape. This point is important for the exam purpose. So oxygen dissociation curve is a S or sigmoid shape. Okay? The next one, going about this one. What I said, this is a standard ODC, that is the SR sigmoid shape. Okay. What I said, what is the importance of ODC? I said different types of factors like temperature, pH, oxygen, carbon dioxide, how these factors influences the standard ODC. Okay, when these factors get changed, either this standard ODC, either it will shift towards the left, or this standard ODC shifted to the right side. Your understanding? Means here with respect to the standard ODC, you can see the two shift. If the standard ODC towards the left shift, towards the right. What is importance of the left shift and the right shift from the ODC? Standard ODC means, now here I am writing the left shift. For example, standard ODC shifted towards the left side. What is importance of left side, you know? That is more affinity. More affinity between what? More affinity between the hemoglobin and oxygen. More affinity means what? There is a more chance of association. Okay, more affinity means more chance of association or more chance of the formation of oxyhemoglobin. So left. What are the factors influences the standard ODC towards the left side? That means, as we know, high PO2, high partial pressure of oxygen, low PCO2. Okay, now already I said these conditions. Next one is that low temperature. Okay, the next one is that high pH. These are the factors. These factors, high PO2, low PCO2, low temperature and high pH. These factors makes the standard ODC towards the left side. Left side means what? More affinity between the hemoglobin and oxygen. If standard ODC shifted to the right side, here I am writing this is the right side. Right side. What is the indication of right side? If the standard ODC shifted towards the right side or right shift. Right shift means less affinity. Less affinity. Less affinity means what? More chance to get hemoglobin get dissociated from the oxygen. So dissociation. Okay. Now what are the factors influences the standard ODC towards the right shift? Okay. That means here low PO2, high PCO2, exactly opposite here. High temperature. Okay. Next one is a low pH. Low pH means what? Automatically high acidity na meaning. High acidity or high H plus concentration. These are the factors makes the sigma, that is a standard ODC, towards the right shift. That is why you need to remember this point. Standard ODC, it influences either left shift or right shift. Left shift, if the standard ODC is shifted towards the left side, that indicates that hemoglobin is more affinity towards the oxygen. If it's a right shift, hemoglobin is more chance to get dissociate. So that means less affinity. These are the factor influences that left shift. These are the factors influences towards the right shift. Now I am going to tell different factors, like I said, the temperature, pH, carbon dioxide, how these factors influences this standard ODC. I will tell you effect of temperature on standard ODC. Okay. You have to concentrate on. Okay, na? For example, first you take the standard ODC. Okay. PO2, I already said, percentage of hemoglobin saturation. Okay, na? Hemoglobin saturation. Standard ODC, I am writing with the black color. This one. It is standard ODC. Standard. Okay. Now, I said a right shift and a left shift. Na? With red color, I am writing this one like this. With another color, to understand for you, this one. Now I am asking you, this right one, effect of temperature on standard ODC. Okay, na? This right one, is it a left shift or right shift? Definitely this is a standard ODC. This is a left shift. This is a, this blue one is a right shift. This means this is a right shift from the standard ODC. This one, that is a red one, is the left shift. Okay, now you are following. Left shift. Now I am asking you. So this standard, what is the temperature? The standard one is a 37 degrees centigrade, as we know, that is a body temperature. Okay. Right shift means, is it a high temperature or low temperature? Definitely right shift means what? Right shift means what? Right shift means more chance of dissociation or less affinity between the hemoglobin and the oxygen. When it is possible, when it is the temperature is high or low? Definitely right shift indicates the high temperature. This indicates the high temperature. Means high temperature makes standard ODC towards the right shift. High temperature means you can take the 42 degrees centigrade. What about the left shift? Left shift means low temperature or high temperature? Definitely that is a low temperature. Means lower than the 37 degrees centigrade. That means when the temperature is higher than the body temperature, standard ODC shifted towards the right side. If the temperature is lower than the body temperature, the standard dissociation curve become a left shift. First you need to understand what is the left shift and right shift. Left shift means what? More affinity. Right shift means what? Less affinity between the hemoglobin and oxygen. Next, a similar way for your understanding. Similar way I am going to tell about the effect of pH. How pH is influencing the standard ODC. Same thing I will draw first standard ODC. Okay. Consider this is the standard ODC. Okay. Now with red color I am writing the left one. This one. Okay. With blue one I am writing this one. Okay. Standard ODC, it is about the influence of the pH. Now, what is the blood pH? That is the standard, that is a 7.4. Now, first left shift. Left shift, when you told pH is a high pH or no, is it high pH or low pH? Or is it a high acidity or low acidity? Definitely you can call the left shift is a high pH. So high pH means what? High pH, that is a pH. For example, what it means? Concept I will tell you. When we start, normally that pH is remains 7.4. Okay, na? When person started doing the exercise, when the person started doing the exercise, muscles need more oxygen. Okay, as a result of the greater cellular oxidation, great amount of carbon dioxide will produce. Okay, right? As we know, carbon dioxide react with the water to form the carbonic acid, that is H2CO3. Then when it get dissociate, it become a bicarbonate HCO3 minus, that I will explain later, plus H plus ions. That means person started doing the more exercise, what happened? Greater quantity of the carbon dioxide. Na? When the carbon dioxide concentration is increasing, it is directly proportional to more H plus ions are producing. Na? More H plus ions means what? That means acidity is a more. Na? So that acidity is a more, means pH is a less. So here right shift I am telling. So first that is a pH is a, sorry, color changed, that is a less pH. Less pH means greater acidity. You are understanding the concept. Greater acidity means what? Greater H plus concentration. When person is doing the exercise, more carbon dioxide you understood, then more H plus ions you understood, greater the acidity. That means the person need more oxygen. That means more amount of oxyhemoglobin, that hemoglobin easily should dissociate it from the oxygen. Then oxygen is more availability to the tissues. That means when this H plus ion concentration or acidity is increasing, automatically H plus ion concentration is increasing, that pH is decreasing, automatically standard voltage is shifted towards the right side. Right side means what? That is a dissociation only possible. Same thing opposite, that is high pH. High pH means what? Low acidity, lower than the standard, that is a 7.4. That is why, see, when the pH is a high, standard voltage is shifted towards the left side, more affinity. That means hemoglobin is not interested in dissociating from the oxygen. When the pH is getting reduced because of the metabolic activities, then standard voltage is shifted towards the right side. Means hemoglobin don't want to associate it with the oxygen. Why? Because more oxygen is required by the tissues. Same way I will tell you, for example, same concept. Now I am telling you effect of carbon dioxide, same way you will get on standard ODC. Consider this one as a standard ODC. Okay. Now this one right shift. This one left shift. Red one is a left shift. Now I am asking you first to take the right shift. Right shift means high CO2 or low CO2. Definitely you can understand right shift means less affinity. Is it less affinity or more affinity? Less affinity, na? That means this is a high CO2. Greater the CO2 concentration means greater the cellular respiration, thereby greater the cell's activity, thereby cells requires more oxygen, thereby hemoglobin wants to dissociate it from the oxygen very easily. Left shift means what? That is a less CO2. That is a way of understanding. Means here, these are the temperature, pH and effect of carbon dioxide on standard ODC. That is why I said in the introduction itself I said the importance of oxygen dissociation curve, you need to understand how the different factors influences association between the hemoglobin and oxygen, whether more affinity between the hemoglobin and oxygen or less affinity between the hemoglobin and oxygen, you can easily understand depending on the condition. Means whether the carbon dioxide more or less, whether the temperature more or less, whether the pH is more or less. When you understand the condition, automatically based on this one, you can easily understand whether the hemoglobin is going to dissociate it from the oxygen or whether the hemoglobin is going to associate it with the oxygen. So these are the different factors that influences the standard ODC. Different factors, effect of different factors on standard ODC. I am going to tell one important thing is a P50. What is actually P50 means? Partial pressure of oxygen where hemoglobin is a 50% saturated, means a half saturated. What is importance of P50 value? You know, P50 is inversely proportional to affinity of hemoglobin towards oxygen. You can understand. Partial pressure, P50 means the partial pressure of oxygen where hemoglobin is a half saturated. If you know the P50 value of certain proteins, where those proteins combines with the hemoglobin, that is inversely proportional to the affinity of that molecule towards the oxygen. I will make you understand. For example, you take P50 value of HbA. What is the meaning of HbA? Hemoglobin in adults, that is alpha 2 beta 2. Okay, now this already I said that. That's P50 value of HbA is 27 mmHg. With one thing you cannot understand whether it's a more affinity. Next I am giving P50 value of HbF, hemoglobin in fetus. That is alpha 2 gamma 2. That's the difference. Here beta 2, here gamma. That is P50 value of HbF, hemoglobin in fetus is a 19 mmHg. Now I am asking you, this is a HbA is affinity towards oxygen, HbF also affinity towards oxygen. Now among these Hb and Hbf, which one is more affinity towards oxygen? Definitely P50 is inversely proportional to the affinity towards. So which one is having the less P50 value? That is Hf na. That is why among these two, which one is having the more affinity towards oxygen? Definitely Hbf. That is hemoglobin in a fetus. Hemoglobin in fetus is having a greater affinity towards oxygen than Hba. With this, what you need to understand? How fetus is acquiring the oxygen from the mother. Means when the Hba carries oxygen towards the placenta, because HbF having the more affinity towards oxygen, automatically it will attract. HbF attracts or takes the oxygen from the mother. That is why fetus is getting the more oxygen from the mother. If you draw the graph, that is a standard ODC. For example, this is a HbA. HbF if you want to write, HbF is a left to the HbA or right to the HbA. Immediately, this is a more affinity. More affinity means left shift. That means you can say like this. This one is a HbF. Why I wrote left this HbA? HbF I wrote left to the HbA. Why? Because left side indicates the more affinity. To understand this one, among these two, HbF having the more affinity. I will give one more example. P50 value of myoglobin. This is also can combines with oxygen. That is 3 mmHg. 3.5 or 3 mmHg. See how much difference is there. Comparatively, myoglobin having a strong, very strong affinity between the hemoglobin and oxygen. How I am telling? Because partial pressure, this P50 is inversely proportional to the affinity between the corresponding protein with the oxygen. This point you have to remember for the all India exams, NEET exams. Last point I am telling you about the Bohr effect. Important thing, this one, Bohr effect. What is the Bohr effect? It is a effect of carbon dioxide and H plus ions on dissociation of oxyhemoglobin. I will explain. It's a Bohr effect is effect of carbon dioxide and H plus ion on the dissociation of the oxyhemoglobin. What it means that I already mentioned the concept. Under normal condition, 100 ml of oxygenated blood delivers 5 ml to the tissues. Okay, this is the normal. When the person is doing the exercise or stress work, that tissues requires more oxygen. Under normal condition, it is giving only 5 ml. But when the person is doing the exercise, his tissues requires more oxygen. How tissues getting the more oxygen when they require or when they are in the exercise or stress condition? That time when the person is doing the exercise, definitely more carbon dioxide will produce. More carbon dioxide means more H plus ions, as we know. What happened? These CO2 and H plus ions become more in the blood. These CO2 and H plus ion go and binds with oxyhemoglobin. These will go and binds with the hemoglobin, force the hemoglobin to dissociate it from the oxygen. Means when these CO2 and H plus ions are more in the concentration, when they become more, when the person is doing exercise. When these CO2 and H plus ions are more in the amount in the blood, these factors, CO2 will go and binds with the hemoglobin, force the hemoglobin to dissociate it from the oxygen. Thereby more oxygen get dissociated, thereby more availability of the oxygen towards the tissues. That is why this one is called as a Bohr effect. What is the importance of Bohr effect? Bohr effect explains the availability of more oxygen to the tissues during the exercise condition. That effect is influencing by the carbon dioxide. That is the effect of carbon dioxide and H plus ions on the dissociation of oxyhemoglobin. This Bohr effect is important for the NEET exam. Till now, if you understand this concept ODC and different factors, you can like and share this video to your friends and subscribe our channel. If you have any doubts, you can comment below. Definitely I will respond to your questions. Thank you.